Welcome to this comprehensive lecture on the fundamentals of biomolecules. Today, we are diving deep into the fascinating topic of enzyme kinetics factors. We will explore exactly what controls the speed of biological reactions, both inside our own bodies and in large-scale industrial laboratories. By the end of this video, you will fully understand the mathematical and physical principles that govern these incredible microscopic machines. To build our intuition before looking at the math, imagine you are managing a highly busy toy factory. In this factory, your workers are the enzymes, and the toy parts they assemble are the substrates. The speed at which your factory produces finished toys is exactly like the rate of an enzyme-catalyzed reaction. If you want more toys, you need more workers or parts. Furthermore, if the factory is freezing cold, workers move sluggishly. If it is dangerously hot, the factory might catch fire and production stops entirely. This perfectly mirrors how biological systems control reaction speeds. Now that we have a mental picture, let us formally define these concepts. Enzyme kinetics is the detailed study of the rates at which enzymes convert substrates into products. When we talk about kinetics factors, we are looking at four primary conditions: substrate concentration, enzyme concentration, temperature, and pH level. Substrate and enzyme concentrations dictate how often molecules physically collide and interact to form a product. More collisions naturally lead to a faster reaction rate. Beyond just concentration, the environment plays a massive role. As temperature increases, molecules move faster and collide more often, increasing the reaction rate. However, if the temperature gets too high, the delicate protein structure of the enzyme unfolds. This process is called denaturation, and it stops the reaction completely. Similarly, every enzyme has an optimal pH level. Any deviation from this optimal pH alters the electrical charges on the amino acids, preventing the substrate from fitting properly into the active site. Let us visualize how these environmental factors affect our enzymes. On the left, the temperature graph shows a green curve rising as heat increases molecular collisions. However, right after the optimum temperature peak, the curve plummets to zero. This steep drop represents denaturation, where the enzyme is destroyed by heat. On the right, the pH graph shows a symmetrical red bell curve. The peak represents the optimum pH, where the enzyme's active site is perfectly shaped. Moving too far left into acidic territory or too far right into basic territory causes the reaction rate to decrease as the enzyme loses its proper electrical charges. Why do these optimal ranges and concentrations matter outside the textbook? Understanding these kinetic factors is absolutely critical in modern medicine and industry. In the human body, a slight fever intentionally speeds up immune system enzymes to help fight off dangerous infections. In industrial applications, companies design laundry detergents with engineered enzymes that must function perfectly in hot water and highly basic pH environments. Furthermore, pharmacologists use this knowledge to design life-saving drugs that intentionally slow down overactive enzymes. To truly understand how substrate concentration affects the reaction rate mathematically, we use a fundamental model called the Michaelis-Menten equation. This equation captures the biological reality that enzymes can become saturated. Let us look at the variables. V0 represents the initial velocity of the reaction. Vmax stands for the maximum possible velocity when every single enzyme molecule is busy. The variable S in brackets represents the substrate concentration. Finally, Km is the Michaelis constant, which indicates the exact substrate concentration needed to reach exactly half of the maximum velocity. Now that we have our formula, let us apply it to a laboratory scenario. Suppose an enzyme has a maximum velocity of 100 micromoles per minute and a Michaelis constant of 5 millimolar. We set the substrate concentration to exactly 5 millimolar. We want to find the initial velocity. We substitute our known values into the Michaelis-Menten equation. The numerator becomes 100 times 5, which is 500. The denominator becomes 5 plus 5, which is 10. Dividing 500 by 10 gives us our final answer of 50 micromoles per minute. Because the substrate concentration equaled the Michaelis constant, the reaction proceeded at exactly half of the maximum velocity. Now that we have calculated these values, let us visualize the Michaelis-Menten equation on a graph. The horizontal axis is substrate concentration, and the vertical axis is the reaction rate. Notice how the blue curve rises steeply at first. This is where adding more substrate drastically speeds up the reaction. But as we keep adding substrate, the curve flattens out, approaching a maximum limit called Vmax. This plateau happens because all the enzyme workers are fully occupied. Also, look at the halfway point on the vertical axis, Vmax over 2. If we drop a line down to the horizontal axis, it hits exactly at Km. This brings us to another crucial mathematical relationship, the concentration of the enzyme itself. If we want to know the absolute maximum speed of our reaction, we must consider how many enzyme molecules are present and how fast each one works. The formula is beautifully simple. Vmax, again, represents the maximum overall rate. The constant kcat is the turnover number, telling us the maximum number of substrate molecules one single enzyme can convert per second. ET represents the total enzyme concentration. This tells us that maximum speed is directly proportional to the amount of enzyme available. Let us see this linear relationship in action with a biological reactor. We're using an enzyme with a turnover number of 200 molecules per second. Our total enzyme concentration is 3 micromolar. We need to find the maximum theoretical speed. We use our linear formula and substitute 200 for the turnover number and 3 for the enzyme concentration. Multiplying these together represents every single enzyme molecule working at its absolute maximum speed simultaneously. The result is 600 micromoles per second. If we doubled the enzyme to 6 micromolar, the velocity would double to 1200. We have covered a lot of ground today. Let us summarize the most important ideas. First, substrate and enzyme concentrations drive reactions forward by increasing productive collisions. Second, we saw through the Michaelis-Menten equation that enzymes can become saturated, reaching a maximum velocity. Third, we learned that adding more enzyme linearly increases this maximum speed. Finally, we visualized how enzymes are delicate structures that require strict optimal temperature and pH ranges to avoid denaturation. Mastering these 4 factors gives us the power to control biological reactions in both medicine and industry. Great job following along.